Hello everyone, my name is Deja Tillman. Today is March 25th, week 3, and 2020, the year of 2020. We are going to do the bacterial morphology and staining techniques. In this lab, I will be detaining bacterial smears using pure samples from broth inoculated in the previous experiments. So this first one I have is the Staphylococcus epidermidis. What I'm going to do, I have my inoculating tube over here, and I will let it dry. Nice and dry. And then I'm going to take the broth and to sterilize the lid by passing it over the flame. Then I will place my inoculating loop in the broth and collect some of the bacteria. With the bacteria I've collected, I am going to transfer my bacteria to my slide here. In a circular motion. Then dispose of the inoculating loop in the bleach solution. Then with my culture broth, I am going to sterilize the lid and recap. I will let that air dry, and then I will move on to my next bacteria, which is the E. coli. Again, with the inoculating loop, you'll let it dry. Let this dry. I'm going to sterilize the top of this broth. Alrighty, then I'll get a sample of the E. coli. Get a small sample of it, and then I will transfer the bacteria onto the slide. Then with my broth, I will sterilize the lid one more time, dispose of the loop in the bleach solution, sterilize the top, and then place the top back on. Then I will take my last bacteria, which is the Saccharomyces cerevisiae. Take my inoculating loop, or make sure it's dry. Go all the way to the bottom. Make sure, actually, you sterilize the lid first. Okay, the lid is nice and sterilized. Then I'm going to get a sample of the loop and repeat the process by placing the inoculated broth on the loop onto my glass to the slide here. This is a new one. And then we're going to transfer the bacteria and then place aside to dry. Dispose the loop in the bleach solution. Sterilize the cap. And then I'll let it air dry. So the first one I let air dry, I am going to heat fix it. So I'll pass these glasses over the flame about 5 times. So 1, 2, 3, 4, and 5. Then that's heat fixing. Then I will heat fix the next slide. 1, 2, 3, 4, 5. Heat fixing the slides will help the bacteria stick to the slide during the gram staining process, which I will do next. I will get the last slide and heat fix it. 1, 2, 3, 4, 5. And I will place these off to the side for drying, which they already are dried because of the heat fixing process. And with these slides, I'll let them cool down for about 10 minutes, and then I'll start my gram staining steps. So that concludes until the 10 minutes is over, and then I will proceed with the stain. So I'll turn off the video for now, then I'll be right back. These, actually, I'm going to show you guys. These are the gram stainings I will be doing. Perfect. I'll be back. Alrighty, guys. So now I have let my slides all cool for about 10 minutes so that the bacteria can stick to the slides. And now I will be applying the stains. I will place the slide in the staining tray to perform gram stains using laboratory and medical settings called differential stains. The first stain I will be using is the Crystal Violet. I will add 1 to 2 drops of the Crystal Violet and let it sit for 60 seconds. 1, 2. I will let it sit for 60 seconds. And with this dye, it will stain the bacteria purple. The stain is associated with peptoglycan. All bacteria has peptoglycan in the cell walls. So that's why it'll stain the bacteria purple. So a couple more seconds and then we will rinse. Couple more seconds, and I have just regular sterile water from the faucet. And a couple more seconds, almost done here. And then after the 60 seconds, I will rinse it with tap water, and it's gonna run until the liquid runs clear. Okay. My next stain will be iodine. I'm going to apply 2 to 3 drops of iodine to the slide. 1, 2, 3. And then I'm going to wait 60 seconds and once again rinse it with water. With this dye, the Crystal Violet is a positive charged molecule and the iodine is negative. Charged molecule together, they will form a complex called the mordant, which is called the mordant. It helps the Crystal Violet to leak to peptoglycan and gram-positive cell walls. And it's because they have a much thicker layer of peptoglycan. So a couple more seconds till the iodine is rinsed with water until clear. So wait 60 seconds. We're almost there. And then almost there. Couple more seconds. And then I will then again take the sterile water and rinse it till the water runs clear. So we're going to remove it now. 60 seconds is up. Let it run clear. Okay. My next stain will be the decolorizing solution. This is the third stain. I will add 2 to 3 drops to the stain. 1, 2, 3. With this one, I only put it for 30 seconds. It's a decolorizing solution. It's a form of alcohol that helps the crystal violet and iodine wash away from the cells that aren't strongly appearing. These are gram-negative cell walls that have a very thin layer of peptoglycan. It leads the crystal violet and iodine in gram-positive cell walls, which have a thicker layer of peptoglycan. And then it's 30 seconds, so I'll rinse it off with tap water. Perfect. After the decolorizing solution, gram-positive cells remain purple crystal violet dye, and gram-negative cells will be colorless from the solution. Then my final stain will be the saffron. I will add 2 to 3 drops. 2, 3. Then I'll let it sit for about 30 seconds. This stain is called a counter stain. It's a pink color. It helps distinguish further between the two cells. After the saffron, crystal violet will still stay in the cell walls, and then the gram-positive cell walls will be all purple, whereas the gram-negative cell walls have taken the saffron stains, making them a pinkish color. So it's 30 seconds. Now I will rinse. Perfect. And then it is nice and rinsed. So now I will let it dry, blot it dry, and I will be performing these stains to all my bacterias, and they will be ready to be viewed under the microscope. This completes the second exercise of week 3 lab experience. So I'll just show you guys my setup really quickly here, and then I will end the video. These are my 3 glasses, and my stains are here. Thank you guys for watching, and I hope you guys are having a wonderful day.